Hi, how are you doing? Welcome to my lab. My name is Dr. Lior Mayo, and this is my lab. This is the part where my students work. This is where we do our actual experiments. And over there, that cool separated room, is where we do our thinking, as you can see by the writing on the walls. And this is also a place when we work with cells that has to be sterile and under very specific conditions. Okay, so what do we actually do in our lab? So in my lab, we are trying to understand how diseases in the brain develop and how we can affect them. For example, we can look at diseases like multiple sclerosis or brain tumors or Parkinson disease, and we try to understand the mechanism behind it. So by understanding the mechanism, we can actually treat those diseases. Now, the interesting thing about brain diseases is there's an interaction between the immune system of the body and the cells that are found within the brain. And that interaction can actually drive disease progression. For example, in multiple sclerosis, the immune system is hyperactive and it actually attacks the brain. On the other hand, when we have a brain tumor, which the immune system is supposed to attack, we have the opposite. The immune system does not respond to the tumor itself. So in my lab, we're trying to understand why and how can we modulate that. For example, one of the things we recently found is that a specific cell in a brain tumor that is native to the brain and can actually affect the way the tumor grows. And by affecting some of the function of that cell, we can eliminate brain tumors completely, which is kind of mind-blowing, to be honest. Another finding that came from our lab is actually a very specific marker for cells from the immune system that goes into the brain. By finding that marker, basically, what we can do now is design therapies that will target those cells, like very specific missiles that will be guided to exactly where we want to go. So we can target the cells. We can use those missiles to actually deliver therapies directly into the brain where they actually need to go, which can really improve the way we treat cells, treat diseases, and minimize the side effects. We can also design new therapeutic methods by doing that, because now we have a very, very powerful tool to do that. So basically, in my lab, we try to do the basic questions and bring them to a phase we can move them into the clinic. And indeed, we have collaborations with companies like Teva Pharmaceutical and other companies really trying to bridge that gap. Let me take you on a tour and come with me into our tissue culture room. So this room is actually when we work with extremely sterile and sensitive cells and reagents. What you see are those things that actually filter all the air coming into the system, so the students can actually work in a very sterile condition and grow the cells they need to do and do the experiments they are doing to manipulate cells and to test our hypotheses. And we have some other equipment here like microscopes and tissue culture equipment to grow the cells and to keep them under very specific conditions in terms of temperature and pressure and oxygen and whatever. And let me show you another thing. This instrument, for example, is one of the key cores of what we do in our lab. This is called a fax sorter. It's an instrument that costs about $500,000. And what it can do, it can isolate specific cells from a population of cells. For example, if you want to study what is happening to a tumor within the brain, we have to isolate the specific cells we want to look at. We can't just look at the entire brain. We need to understand what a specific cell does. So these instruments actually allows us to do that. And what you can see is that we can start from a very, very big group of cells, and every dot here represents a cell. And by a very specific and very unique system, we can go down to get a very, very, very specific cell population. And then we can study it, we can study the effect, and we can study how the different therapies we are trying to do and design actually affect what we want to do. And that is really an amazing piece of technology that really helps us to move our research forward. What we are doing now and what we want to do. So what we are actually trying to do in our lab is to grow this program even more. For example, while now we treat a very specific type of brain tumors, what we will also like to do is to develop another program to treat brain tumors in kids, which is a completely different mechanism and completely different questions, although we think some of our findings from the adult brain tumor can actually be also implicated in the kids. So this is a program we're trying to build, and of course we have to raise funds to do that. I'm estimating a few $100,000 to actually do that. Other things we try to do, for example, is to take the information we gained about the marker from the specific immune cells and look how it can affect other diseases. So we already found a correlation with brain tumors. We found a really an amazing correlation with Parkinson's diseases. Apparently, mutation in this protein, which is not expressed in the brain, are associated with developing Parkinson's disease. So now we want to understand exactly how they work, and if you can use our knowledge about that marker to actually better treat Parkinson's disease and better diagnose Parkinson's diseases. So that would be really, really amazing. And of course, for that, we also need, of course, funding for the students. We need funding for the research program. We need funding for different instruments we need, like very high-resolution microscopes and other pieces of analyzing of specific cells and what the cells do and their phenotypes. So actually, sadly, it all goes down to money in the end. What we can do also affected by our ideas, but it's also being affected by the amount of funds we have to do that. So any help you can offer will be muchly appreciated.